Next up, we have Professor Tom Brown. So during the week, see Professor Tom Brown is a professor of chemical biology at the University of Southampton. You can see behind me that on the weekends, he's Iron Man. He's founder of 3 biotech companies. So it made me wonder if one of those is Stark Industries. I don't know. Okay. Anyway, he's a fellow of the Royal Society of Edinburgh and the Royal Society of Chemistry. And please welcome Professor Tom Brown.
Okay, I'd like to start by thanking the organizers for giving me the opportunity to speak at this really exciting meeting. I was here 4 years ago at the Molecular Frontiers Symposium in Singapore. It was a very enjoyable experience then, and this is just as good. Thank you. Today I want to talk about, let's let the slides come up actually. Yeah, today I want to talk about some of the research that we're doing in our lab, but I want to also talk a little bit about ideas, about how ideas come in research and how we can generate new ideas and new theories. And I want to start by talking about this book by Richard Dawkins, who's a very well-known author and a strong supporter of Darwin's evolution theory. There was a cleric, William Paley, who gave evidence for a creator by saying that a human being couldn't be created, had to be created, because a human being is a very complicated thing, just like a watch. It has to be put together very carefully, it has to be designed, and that requires a designer or a creator. And Dawkins contrasted this view by saying that, but this is not a random process. We have an environment and we have evolutionary pressure. So you can have a blind watchmaker who can't see what he's doing but moves things around, shuffles things around. He makes things, and these things are selected by the environment to be good or bad. So you don't need a creator, you just need some kind of random process with pressure, with evolutionary pressure. Now, I'm not here to talk about religion or to think about whether God exists or not. That's not my role today. But it's interesting to think about how to generate new ideas and how to generate new concepts in science by different methods. Now, one of the best examples given by Dawkins is the eye. And just to explain that the idea is that you don't have to have a creator to make something as complicated as an eye, because what you can do is you can start with a very primitive eye. You can start with something really simple that doesn't work very well at all. But if you're the only organism that has an eye, you have an advantage. So imagine that you're a worm swimming in the sea, a little aquatic worm, and the sun's shining down, and you've got 2 little eyes that work a little bit. And every time a predator comes along, he casts a shadow. So you swim away from the predator, and you don't get eaten, but all your friends get eaten. So straight away, an eye, a very primitive eye, is a fantastic thing to have. It doesn't have to be as sophisticated as a human eye, but you can very easily follow the process by which a very simple primitive eye, which is just a light sensor, develops over an evolutionary time scale and becomes a proper functional human eye with a lens, a cornea, an iris, a sensitive retina that can see different colors. So evolution, natural pressures, can give us these very complicated machines, really. So now, as scientists, we have options how we progress with our science. And that will depend upon the nature of the project and the complexity of the project. In chemistry, we often apply creative design. We generate molecular models, we write on pieces of paper, we design things which we think will do the job, we make them and we test them, and then we improve on them. And that works very well in chemistry many times. In biology, often we can't do that because the system is extremely complicated and we don't understand it well enough, so we have to have a means of generating some variability, applying some pressure, and creating something that's better than our original version. And biologists have done this kind of thing for many, many years, and it's led to amazing advances. We've seen already in this symposium some great examples of how biology has advanced. A couple of examples that we can see here. We can make entire genomes now. Craig Venter beautifully explained how we can do that. We can generate all sorts of new types of organisms through molecular biology which do jobs for us, which make things for us. But we have to remember that enormous impact has been made by chemistry in all these areas, and physics. A couple of examples are this GFP, the fluorescent proteins, and methods of DNA sequencing. So the contributions from the logical design of machines and processes by chemists and physicists have played a very important role in biology. And this is something that we must always bear in mind. Now, the way that we can use evolution to our advantage in biology is we can speed up evolution. We can make it go much faster. There are many different types of biomolecules that we can generate. A good example is aptamers. Aptamers are these molecules here, DNA or RNA molecules, which bind to proteins or other factors and do a particular job for us. Now, these are far too difficult to design at the molecular level. They're too complicated. But if we've got a process by which we can mutate genes and shuffle genes around, and if we have a reporter system that tells us whether we've made something better than the previous example, and if we have a cyclic process by which we can select a good example, mutate it further and improve it, we can use evolution. We have a blind watchmaker which can generate for us very important biomolecules that can do therapeutic jobs, like, for example, aptamers to prevent blood clotting or to attack cancer cells. And we can apply directed evolution to change the enzymes that are in our bodies, like DNA polymerases, and we can make them behave in different ways. And I'll come back to that later. But as long as we've got this process, this cyclic process of generating, selecting, and selecting again, we can do that. And of course, nucleic acids are the molecules that do that job. I want to just briefly mention the emerging field of synthetic biology, whereby we can actually take advantage and take command of molecules in cells, and take command of cells, and we can generate new types of cells, new types of organisms, and we can use the cell as a reaction vessel. I'm not going to talk about this in great detail, but just to mention that this is a very important emerging area, which is going to be incredibly important in the next 20 years. And it demands a command of biology and of chemistry. I want to just change the topic for a minute, and I have a question. The question is, what do these two pictures have in common? We have a picture of the Americas, and we have a smiling face. Very different things, but they've got something in common. So what these pictures have in common is DNA. These two pictures are made of DNA. They're made of DNA origami. DNA origami is a process whereby we take a very long piece of biologically derived DNA. It's actually coming from a virus, and it's single-stranded. And we then take lots and lots of very short pieces of DNA, which we make in the lab. We mix them together, and the small pieces of DNA act as staples. They staple together the big piece into a particular shape. And a man called Paul Rothermond has developed this technology, and you can use it with the aid of a computer program to generate objects, nano-objects made of DNA. And you can image them by atomic force microscopy or similar kinds of techniques to produce a nano-skater, nano-china, nano-boxes, and within these nano-boxes you can put molecules, and you can open the box and you can close the box. Now, this is a very fascinating area, and it's blossomed, and you can imagine that people throughout the world are logging into these websites, designing their nano-objects, and painting beautiful pictures with DNA, using DNA origami. Now, the kinds of pictures that they can generate also include the ones I've shown here. These are all DNA on nano-objects on surfaces, as is this object, a polyhedra, and this tetrahedron. These are all made of DNA. Now, a few years ago, we started to carry out a research project in my lab in collaboration with Professor Norden, who's in the audience, and he's, of course, he's the main driving force for this particular initiative. And the idea was to use DNA as a scaffold to build molecules onto DNA and use DNA as a benign scaffold at the nanoscale to do this kind of thing. So, it's a great molecule to do this for various reasons. It's easy to make, to synthesize. It self-assembles in a predictable manner. It's a very stable molecule, and it can be very easily attached to surfaces. So it's got all the right characteristics. And the work that we did with Professor Norden was to do a bottom-up approach to make very tiny nanoscale DNA objects, like the one shown here. This is six different DNA duplexes with arms. This is the object in one direction. This is the same object rotated by 90 degrees. And the idea was that we could take such objects and we could build them into bigger nano objects in a genuine bottom-up, entirely synthetic approach. And what I mean by synthesis now is chemical synthesis. That everything in this molecule here has been synthesized on a machine. It's a piece of DNA. So this is a purely chemical synthetic approach, bottom-up approach to nanotechnology. So the idea is that you generate specially shaped DNA molecules, which happen to be Y-shaped DNA molecules, and every one of them is unique. And they can only assemble in one way to produce a nano object. This is our nanonaphthalene, and this is our nanoanthracene, entirely made of DNA. So it's a bottom-up approach to build molecules from DNA. And we were able to image these objects using atomic force microscopy to produce images like nanonaphthalene, and you can see the two rings here. And this is only about 10 nanometers in total size. Nanobenzene here. So we were able to use this approach to make these objects. However, there is always a problem when you're using DNA to self-assemble, because we have good guys and bad guys. The good guys are the Watson-Crick base pairs, the AT base pair and the GC base pair. And these are the guys that enable us to assemble things in a predictable way. But we have 6 bad guys. We have lots of mismatch base pairs where the bases have come together incorrectly. And that means that the DNA folds incorrectly. So as soon as we start to try and build up these objects, we have a problem that 99% of the time everything works fine, and then occasionally one of these bad guys creeps in and messes up the whole show. So we have to have a means of cleaning things up. And the way that we do that, and I'm going to just talk about, now I'm going to step back a little bit again now, because I'm talking about some chemistry, some specific chemistry now, developed by Professor Sharpless and his colleagues at Scripps, and you're going to hear much more about this chemistry tomorrow, so I'm not going to dwell on this. But I'm going to talk about a chemical reaction between an azide and an alkyne, which is catalyzed by copper-1 and produces a special linkage called a triazole. And I'm going to show you how we can use this chemistry with DNA to do some interesting things. And the reason that this chemistry is good for DNA, there are many reasons, but one reason is that this alkyne and azide, these functional groups, are very specific in the way that they react with each other. They don't react very well with other functional groups. The reaction works very well in water. Many organic reactions don't work in water. This one works very well in water, and DNA needs to be in water. And the reaction can be switched on when we want to switch it on by adding copper. So we can bring all the objects together, make sure we bring them together correctly, and trigger the reaction with a little bit of copper. And the product is a very stable product, and it's not toxic. So it's a really, really nice reaction to do on nucleic acids. And we use this reaction to take these nano objects and to click the sides of them together. So here you can see this is a 6-membered ring. It's a benzene ring, but it's really DNA. And at each corner of this DNA hexagon, we're clicking together the sides, so that when we've generated this nano object, it's a stable object. The Watson-Crick base pairs have been formed, and they can't come apart, and the good guys have won. Everything's fine. And then we can purify this nano object from its crude form into its pure form on a gel, because it's a stable molecule. And this then becomes a building block to make much bigger pieces of DNA. So this was the idea, and the idea worked. But immediately, we decided to think about this in a rather different way. We have a very nice piece of chemistry here that we can work with use on DNA, but can we use it in a biological context? Now this slide shows us an alkyne and an azide, and in between them is actually a very long piece of DNA. If we make this molecule, and if we dissolve it in water and we add some copper, it curls up and it cyclizes and it reacts to form a triazole linkage, and we can do that twice. Unfortunately, one of the colors has completely disappeared, but this is actually a double-stranded piece of DNA which has been made by click chemistry. And this is a picture of it enlarged. It's a little bit like a tiny virus particle. It's a double-stranded piece of cyclic DNA, which has got 2 triazole linkages in it, holding it all together. So it's kind of a model for, if you like, a DNA-like object, a virus-like object. And we started this work about 4 or 5 years ago, and then we went on to play around with this type of chemistry in different contexts. We wanted to find out what is the smallest DNA object that we can make, which has got Watson-Crick base pairs and which is stable. So we took various pieces of DNA of different lengths and we cyclized them. And we went right down to the point where we had 2 base pairs. Now, 2 base pairs doesn't exist in nature as a stable object because the base pairs are not strong enough. But if you cyclize these small constructs in the way that I've shown here, by having a triazole linkage at one end and something equivalent at the other end, you cage them, you put them in a cage. And when you put them in a cage, they're now tame objects, and the base pairs form. And the Watson-Crick base pairs are very stable. So we have 2 base pairs of DNA clicked together using this reaction, this click reaction, and the molecule behaves like normal DNA. The base pairs are stable because they have hydrogen bonds and they have base stacking. Base stacking is very important. They have a normal spectrum, a normal CD spectrum to show that they've got a bDNA helix. They bind to drugs that bind to DNA, so they're very nice models for studying various little pieces of DNA. And this is what they look like in 3 dimensions. Here we have the 2 base pairs. So this is probably the smallest DNA duplex that can exist in a biological environment. These base pairs exist well above room temperature, are very stable. We did the same thing with 1 base pair, and we didn't get any base pairing. When you only have 1 base pair here, there is no base stacking. Base stacking is very important to glue DNA together. So without base stacking, the hydrogen bonding between the bases is not strong, and the 2 bases just drift apart, and there's no base pairing. So 1 base pair doesn't work, but 2 base pairs is extremely stable. Now, having done that, we wanted to see if we could mimic enzymes that stitch DNA strands together. Enzymes that occur in nature to stitch DNA strands together are essential in all living systems. The process of replication, recombination, DNA repair, many biologists depend entirely on the ability to glue together DNA and RNA strands. And the enzymes that do this are called ligase enzymes. And this is a picture of a ligase enzyme which is stitching together 2 DNA strands. Now, ligase enzymes are important. We wanted to find out whether we could substitute chemistry for this biological process. So we have a little bit of a problem here, because as chemists, there are some things we can do and some things we can't do. We wanted to design a linkage, a DNA linkage, that would mimic a normal phosphate bond between 2 DNA strands. So we had to start with something that we could make in a reasonable time scale. We only have a certain amount of time to do this, unlike a biological process which occurs over millions of years, evolves. We wanted to do this in a short time scale, so we designed this linkage here between 2 DNA bases, and this linkage would be used to join together 2 big DNA strands instead of using a ligase enzyme. Now remember that we're talking about DNA, which is normal DNA, except at one particular point where we join together the 2 strands, where we have this different linkage. So we carried out the chemistry, and this is just to show the linkage, the modeling of the linkage. So the linkage itself looks a little bit like a normal phosphate bond, but in detail it's very different. It's longer, it doesn't contain phosphorus, and it's not charged. So it's very different from a normal phosphate bond, but it does allow the DNA strands to have base stacking, which you can see here in 2 different orientations, just like normal DNA. So if you step back a little bit from the close-up, you can see that this linkage can allow the DNA to behave normally. And we were able to prove that we could click together 2 DNA strands using this chemistry and get a pure product, which is shown here. And that product would itself be amplifiable by enzymes that amplify DNA. this is using the polymerase chain reaction, or PCR, we could actually take a DNA template containing this unusual linkage in the middle, and we could read through it with DNA polymerase enzymes. Now, this was a surprising result, because this is such a different linkage from normal DNA. We really expected that the polymerase would stop when it saw this linkage, because in an evolutionary context, DNA polymerases have never seen this kind of unusual linkage. However, they have seen damaged DNA. Remember that DNA polymerases in our bodies, in our cells, have got to be able to read through damaged DNA, because all the time the environment is causing damage to the DNA in our cells. And if that DNA damage was not repaired, we wouldn't survive. Our cells would die. So DNA polymerases have never seen this kind of linkage, but they've certainly seen lots of damage to DNA. So maybe it's not so surprising that they can rescue the situation when they see something unusual. Anyway, they can do it, and we prove that. And this is the band. I'm sorry, I've gone too fast now. It's gone over at a tremendous pace, but here we are. Okay. So this is the band of the product of this PCR reaction containing the unusual linkage. It works. So we've taken a click reaction to join together two DNA strands to produce a product which has got the triazole linkage in it, and we've amplified it by PCR. But that doesn't tell us if we've amplified the correct sequence, because in all living systems, this is not going to be useful unless the correct product is produced. In order to find out whether we have the correct product, we have to sequence the DNA. So we took the DNA and we cloned it into plasmids, put it in bacteria. We read the DNA sequence that was produced by reading through this linkage. And we found something slightly perturbing, slightly disappointing. We found that the enzyme had jumped over one of the two bases at the ligation site. It had ignored one of the two bases. It had not produced a faithful copy. It had, in fact, at this linkage site, ignored one of these two T bases. So this sequence that was produced was one nucleotide shorter than the correct sequence. The enzyme basically looks at this template, it sees this thymine, but it doesn't see this thymine, and jumps to the next nucleotide. It had rescued a bad situation, as it has done throughout evolutionary timescales, making something so that the cell can survive, but not quite making the right thing. Nevertheless, this is a very interesting thing, that you can read through these unusual linkages in DNA and get some functional product from it. But we wanted to design something which would work better, ideally perfectly. And again, we're limited. We only have a short time. We only have a little time to do this. We can't rely on evolution, really. So we designed something next which we thought would be a better mimic of normal DNA, for which we had the chemical tools to synthesize. And now we decided we maybe need an oxygen here, like normal DNA, and maybe we need a CH2 here, like normal DNA. So we designed a different linkage, which is this one here, which we're hoping will be a better mimic of normal DNA. And again, we use this linkage to join together 3 DNA strands to put these triazole linkages in 2 places. We carried out PCR amplification, and we carried out DNA sequencing, the kind of standard protocols. The PCR worked perfectly. As you can see here, this is the product of the amplification of this DNA. And although we can't see much on the slide, this is actually the results of the sequencing. The result is that everything works now. This is a mimic of normal DNA, which polymerase enzymes can read through perfectly. No longer do they jump across the linkage, they read through the linkage correctly. So we can now do pretty much, we can now use this as a proper DNA mimic. That means, in principle at least, that we can use this kind of chemistry, rather than biology, to synthesize genes. We can take several strands of DNA with these linkages spread out, we can put a template or splint against them, and we can stitch them together purely by chemistry, no enzymatic involvement. And we can also do the same with cyclic DNA. Now, this is work in progress. This is not the full story. And indeed, it would be foolish to try and mimic the achievements of people who've been making genes and genomes in biology for many years and try and, this is new technology and it's purely chemical. But in principle, we should be able to have a completely chemical approach to at least inserting pieces of DNA into genomes. Second question, can we actually transcribe through these linkages? Replication is DNA making DNA. In the central dogma of molecular biology, DNA makes RNA, makes proteins. Now, we need to, if we want these linkages to be functional in living systems, they need to be able to be read through by RNA polymerases, molecules which convert DNA to RNA so that RNA can make proteins. So the test is, can we take one of these DNA templates and can we read through it with an RNA polymerase? And the answer is that we can. This is the picture. Again, this is a schematic. The DNA goes into the RNA polymerase, and out of here comes a single strand of RNA. And this is the evidence by gel. This is normal DNA template, and this is a triazole DNA template, producing RNA from that linkage. Here we have a very simple experiment where we read through the DNA in a linear way as opposed to PCR. It's very similar to what we did before, but it's further confirmation that the linkage can be read through very quickly. This is normal DNA and this is triazole DNA. So polymerase enzymes, whether they be DNA or RNA polymerases, can read through these linkages and make a product. This is all in vitro. This is in a test tube. This is biochemical transformations. What about biology? E. coli is the standard lab bacterial organism that people use throughout the world. We took our click linkage, inserted 2 click linkages into a plasmid in an essential gene which confers resistance to antibiotics. We inserted the plasmid into E. coli and we grew colonies of E. coli. Now the idea is the colonies can't grow if the antibiotic resistance gene is not present. Because when we treat in the standard way the bacteria with antibiotics, unless our triazole linkage is functional, the bacteria will be killed by the antibiotic. And what we find is that we get normal colony growth with our triazole DNA. So we can now say that this linkage is biocompatible because within E. coli it's treated as normal DNA. We can even take a strain of E. coli that can't do proper DNA repair. When we take a strain of E. coli that cannot do proper DNA repair, it still is able to survive in the presence of the triazole linkage. So the linkage is not being repaired, it's being used. It's being functionally used rather than repaired. So we have a system which works in bacteria. A linkage which behaves as a normal DNA linkage in bacteria, at least to a first approximation. What I mean by that is that the rate at which the enzyme goes through the linkage is a bit slower than normal, but it goes through normally. And we can try and rationalize this. As chemists, we can try and work out what the real structure is that we're working with. And we do that by NMR. We make a piece of DNA containing this linkage, we put it in a test tube, and we study it by NMR. We look at the structure of the DNA containing the linkage. And this is a DNA duplex that we have here, and the DNA duplex contains a triazole linkage. The triazole linkage is shown here in green, and as you can see, the blue structure is the DNA containing the triazole linkage, and the orange structure is DNA which is normal DNA, which does not contain the triazole linkage. And they overlay very well, they look very similar, but at the point where we have the triazole, there's a little kink in the DNA. And the reason there's a little kink is because the backbone at the triazole linkage is a little bit longer than normal. So in order for the DNA to preserve its normal B-DNA helix, its right-handed B-DNA helix, there has to be a little kink here. So the little kink that we have there is potentially a problem, but doesn't seem to worry DNA polymerase enzymes. But again, this is unfortunately not coming through very well. But intelligent design would tell us that we would be better off with a slightly shorter linkage, because the linkage that I've just shown you has a small kink. So we designed a linkage here, which you can't really see, unfortunately, which is the same length, the same number of bonds as in normal DNA. It's a little bit more difficult to make, so we didn't make it until more recently, but it looks on paper to be a better mimic of normal DNA than the one that we produced. So we went on to make this linkage. The click chemistry worked extremely well. The PCR reaction worked extremely well. It's looking really good now. Intelligent design we have here. We make the right linkage, the right length. I'm not going to show you the results of the sequencing because we got lots of results from the sequencing, very few of which were correct. This linkage is not read through properly by DNA polymerases, and RNA polymerases just look at it and drop off. I thought it was an intelligent design, but in reality, our Mark II version, the one that worked well, is much better. So clearly there are many factors which influence the design of a linkage that will work. We can rationalize it in many ways. The nitrogen atoms of the triazole hydrogen bond to the polymerase enzyme and happen to fall in the right place to do that. Whereas this shorter linkage is a little bit more rigid and probably the nitrogens don't quite fall in the right place. For the last thing I want to mention just briefly now is that we've also looked into the RNA world with our artificial DNA linkages, artificial linkages. I think most people are familiar with the pioneering work of Andy Fire and Mello, who discovered RNAi, interference RNA, and were able to show that small RNA molecules can influence gene expression. Genes can be switched on and switched off by small RNA molecules that are generated in cells. So the RNA world is a fascinating world, and I don't have time to talk about it in detail, but if you have time, you should read about the RNA world and the new developments in microRNA, siRNA, and all these various small RNA molecules which control the expressions of genes inside cells. But just to cut a long story short, we wanted to see if we could use our click chemistry developed from Professor Sharpless's original work to build big RNA molecules from small RNA molecules. The chemical synthesis of RNA is actually very difficult, much more difficult than DNA synthesis. So to be able to build up RNA from units, from small units, is very important. So we were able to demonstrate we could take small pieces of RNA and we can label them with fluorescent dyes. We can mark them. We can mix them together and they will self-assemble into a structure which can then be clicked together to stabilize it by making chemical bonds across the bases. So we can click together RNA in the same way as we can click together DNA, but we have a real big advantage now because RNA can form tertiary structures which will enable us to template the reaction very nicely. So the reaction works extremely well for RNA, and we can generate catalytically active RNAs which cleave their substrate molecules, and they're completely functional. So the same kind of linkage, this is just to show that we can assemble, this is a crude RNA made by click chemistry from 3 pieces. It's just a single product. And that RNA molecule cleaves its substrate very quickly at room temperature, very cleanly, just like a normal RNA molecule. So we can assemble RNA chemically very efficiently. I just want to finish by explaining some of the ideas that we have for future work. And to come back to, this has so far been chemical design and rational chemical design, although some of it wasn't so rational. But I just want to mention one idea that we have. If you have a piece of DNA or RNA and you put it in vivo, if it has a triazole linkage, it's completely different in that particular facet from all the DNA in the cell. So maybe we can put our artificial DNA in cells with the triazole linkage in one position and evolve an enzyme that will only recognize the DNA which has a triazole. So maybe we can develop enzymes which will switch genes on only if they contain a triazole linkage. So we start with a normal enzyme, which is working on normal DNA, and we evolve from it an enzyme that prefers a triazole linkage. And just to mention why we think this is feasible, you can sequence the DNA from a woolly mammoth. You can dig up a woolly mammoth if you can find one. You can thaw it out. You can take the DNA from it, and it's all broken up in small pieces. But you can sequence it, and you can determine the sequence of the woolly mammoth. This has been done. particularly well if you use a polymerase enzyme that's been evolved to deal with damaged DNA. So we know that we can change the structure of a polymerase enzyme to do things it couldn't normally do. So the idea is, let's take some enzymes that work on DNA and let's see if we can evolve them into doing jobs on modified DNA. Finally, I just want to mention my colleagues in Southampton who have been involved in carrying out this work, who are shown here. The majority of the work has been done by Afaf El-Sagir here. Also to mention, the work has been partly sponsored by a company called ATD Bio, and if you're interested in finding out more about nucleic acids, learning more about nucleic acids, this website here has an online book which has chapters on all different aspects of nucleic acids at the undergraduate level, so it's free to log on and read about nucleic acids. And finally, I'd like to thank you for your attention. Thank you.